the microbiome, a healthy microbiome, effectively underpins overall health in the animal. And that is because the microbiome interacts with or is part of the immune system in the gut, and they also are responsible for metabolizing or fermenting various nutrients and providing metabolites that the gut and the animal itself can use for energy and other processes. So they're pivotal to our health.
Hello and welcome to the Pet Food Science Podcast, where our goal is to share research findings to help support the continual innovation in the pet food and nutrition industry. I'm your host, Dr. Kate Shoveler, and I'm here today with today's guest, Dr. Emma Bermingham, to discuss with us about how diet affects the microbiota and how the microbiota ladders to health. Thank you for being with us today, Emma.
Thanks for having me, Kate. It's nice to be here.
Thank you. As maybe a point of introduction, I just wanted to introduce Emma and her experience. Dr. Bermingham has more than 15 years of experience in the pet food industry. After a postdoctoral position at the Waltham Centre of Pet Nutrition in the United Kingdom, she established a research platform at Ag Research Limited in New Zealand that focused on the impacts of high-protein diets on gut health in both the dog and the cat. During this time, as well as conducting fundamental research, Emma consulted for pet food companies in New Zealand, Australia, Canada, and the United Kingdom. Additional research focus areas at Ag Research include energy requirements of pets, which, by the way, Emma, is a very big focus on my energy section in my pet nutrition course.
Excellent.
The role of the microbiome in pet health, the impact of age and diet on cat nutrition, and how animal-derived ingredients could provide nutritional functionality in both dogs and cats. In 2022, Emma left Ag Research and went to work for industry. Welcome, Emma, and I'm very excited about today's conversation.
Thank you, Kate. So am I. I love talking about cats and dogs.
I just love talking about biology in general and how we meet anything. It's so complex that people really don't appreciate that it takes an entire village. And you've provided a tremendous amount of data, fundamental data in pet nutrition to help guide the industry. So first and foremost, I compliment you on the vast amount of contribution you've done there. So let's talk to the pet food industry about maybe how they can use your data even better. So I thought we'd start just basically, if you would not mind describing why it's important to do microbiome research in both the dog and the cat.
It's a very good question. So we know from data in humans and models of human nutrition, so rodents, pigs, etc., that the microbiome, a healthy microbiome, effectively underpins overall health in the animal. And that is because the microbiome interacts with or is part of the immune system in the gut, and they also are responsible for metabolizing or fermenting various nutrients and providing metabolites that the gut and the animal itself can use for energy and other processes. So they're pivotal to our health. The microbiome obviously exists in many places other than just the gut. We've got a skin microbiome, we've got an ear microbiome, those sorts of things. But our focus has really been around how does diet and diet nutrients impact the microbiome and then have a further impact or potential impact on host health, so the dog and the cat.
Fantastic. So maybe to frame up a little bit more too, let's talk about first differences in the microbiome between cats and dogs? Are they the same? Are they different? How do pet food companies think about supplying those microbiomes differently, if you can pluralize microbiome?
Okay. So we'll take a step back. So I guess if you look at the pet food industry, there's been a real heavy focus on the dog. Dog is the largest market, and therefore a lot of the research that has been done in the pet and pet nutrition has focused on the dog. Cats have basically been forgotten. I'm team cat, so if I have a choice, I'll go the cat because they're awesome little creatures. I guess in a lot of ways, it has been assumed that cats are basically little dogs, but they're not. It's quite difficult to compare directly because cats and dogs actually have quite different nutritional requirements. So if you're trying to understand how a cat and a dog will respond to a nutrient, you've got the complexity. You need to be feeding it a complete and balanced diet. And actually some of those proteins, fats, and micronutrients actually differ between the cat and dog. Some work that we've managed to do through government funding when I was at Ag Research, and also we had the support of the New Zealand pet food industry, we had an opportunity to look at very similar diet formats where we had effectively dogs on a high protein raw meat diet and cats on the same raw meat diet, and the only difference between the two groups was the mineral premix. So there are some differences there. We can't get around that without kind of causing ethical concern. And what we found was that what we're discovering, because we're actually diving really deeply into that data, is that the microbiome between the cat and the dog on similar diets is actually quite similar. And so we're trying to understand what genre, what families of bacteria are producing butyrate and propionate. That's a place we've started to focus purely because of the health benefits of butyrate and propionate for the animal generally. So we're taking a real deep dive into the function of the microbiome, trying to understand, do the dog and the cat respond the same, or are they different? And actually, they're quite similar, but there are some quite key differences which we're starting to tease out now. So I can't give a lot of detail around that, but yeah, the notion that cats are small dogs is certainly not true. And so therefore, research going forward, the cat needs to be the cat and the dog needs to be the dog. There was a nice wee quote that I like to use, and it's only people who love cats that focus on feline nutrition, but hopefully the pet food industry in the future will start to kind of realise that cats are actually important too and need to be researched in their own capacity. So.
Well, and maybe, not the topic for today, but I think the last demographic information I saw is that younger generation, whatever we call the generation
millennials, or
yeah, I think the generation after millennials now, are they Gen Z? I think, but you're starting to see actually a migration towards cat ownership.
Yeah, that's right. Yeah.
Dogs. So if we were to, I too am a cat lover, and I agree. There's such interesting animals from birth through their life, just so unique. But the pet food industry needs to start generating this data at this point because the demographics are changing. And if they want to make sure that they that they formulate and market foods that allow cats to thrive, we need a lot more data.
Yeah, we do. Yeah.
There's hardly any. So little plug for research there on the side. But stepping back to your last comment, so I want to maybe ask then, so the microbiome is largely defined on what a mammal, and I'm just going to put mammals together in a bracket, on what they eat. and maybe a little less so based on species differences. Is that correct holistically?
Yes, I think so. So my journey into the microbiome started in 2013, where I was very lucky to come and go to Illinois and work with Kelly Swanson in his lab there to start kind of understanding the kind of dietary impacts on the microbiome. I'd received some funding and it was good, but it wasn't a lot. So we'd had to settle on feeding commercially available diets. So we had a kibble and we had a canned or a retorted diet. And so we were seeing differences because basically dietary nutrients will drive the profile of the microbiome. But when it came to kind of really understanding what those differences mean, we kind of kept hitting this wall of increased high protein diets increase the level of bacteria that can utilize protein. However, when you look at the literature, these high protein utilizing bacteria are typically associated with disease in human models. So in unhealthy humans, also unhealthy gut, you have blood mucus entering the intestine. Those are proteinaceous. So you get the microbiome, the protein utilizers increase, and therefore there's this correlation that high protein diets are associated with negative bacteria in the disease. Obviously, causation, correlation, we can park that for another day. And so when we were trying to understand our results, it was, high protein diets equals so-called bad bacteria. And from an evolutionary point of view, this didn't really make sense to me. It's like, why would a carnivore who's evolved to consume high protein, high fat, sometimes low fat, depending on where seasonal, the season was, certainly high protein, and how could that be negative to their health? We can park the carnivores in the wild, don't live that long. We'll just park that for the time being. But so it set me on a journey to really understand the carnivore microbiome and what it actually meant. So I spent a lot of time supported by industry, who in New Zealand is high protein and low processed type diets, to really understand the if we see these changes in the microbiome, what do they actually mean? And so so I guess trying to take a few different approaches to really understand the dog as a dog and the cat as a cat, because sometimes they're the same, sometimes they're not, but let's consider them separately. And they're certainly not humans, and they're definitely not rodents and not pigs. And so, really trying to understand, I guess, that whole carnivore context overlaid into the microbiome.
ProAmpac, your companion in pet packaging. Visit pets.proampac.com to explore our innovative sustainable solutions, such as our QuadFlex recyclable flat bottom bags, ProDura polywoven bags, ProEvo recyclable paper bags with grease resistance, and our proactive recyclable film and pouches that run at optimum speeds on your filling equipment. Elevate your wet food, treats, and kibble brand by utilizing packaging that safeguards and preserves product freshness. Trust ProAmpac for packaging that cares for your pets and the planet. pets.proampac.com.
Yeah, so the diet will push changes in the microbiome, but the products of fermentation that the microbiome produced do not have the same effects across all species.
That's
because
correct,
part of it is predicated on their evolutionary diet, and
yes, that's right. Yeah,
that must mean that there's different responses to those. So the other component, it's very interesting. The other component to this that that pet food companies probably need to think about, and my students ask me this all the time as we tried to decide based on life stage
Mm-hmm.
what markers of health are, because, and I'll often juxtaposition my students to me and remind them that they're 30 years younger than me. And so the idea of what will affect their health acutely is far different than what will affect my health acutely. I seem to be much more sensitive now than I was when I was 21, as an example.
Yep.
So when we think about that age and that lifestyle component, and we think about birth of dogs and cats to end of life, how does the microbiome then, when we think about a healthy microbiome, is it universally the same, or do we have to consider development, maintenance, and then probably age-related changes to that? What do we know about that in dogs and cats?
Yeah, so certainly the ability to study the microbiome in terms of that from birth right through to old age is quite limited in a research context, and that's simply because research colonies tend to have a stage at which the animals might be rehomed, for example. At Massey University, so I've done a lot of work with Dave Thomas and his cat colony, and we have looked at, we started with this sort of idea of epigenetic regulation of the cat, effectively, looking at two different diets, the kibble and the retorted diet. We fed those to mums, pre-pregnancy, pregnancy, lactation, and then we followed those kittens right through to, I think the last samples we got off them were about 12 years of age. And now those cats have been retired, and they're living out their lives in the retirement pens at Massey University. And basically, what we see is that there's the strength in this is you can follow the same cat through its life cycle. You don't have that kind of individual variation that you need to consider if you're looking at discrete age groups. And what we see is that the microbiome of the young growing animal is quite volatile. It shifts around quite a lot from sort of birth through weaning to about 1 year of age, and then after that it's relatively stable. You do see minor shifts, and so I guess from a research perspective, it's understanding what those minor shifts could mean. We've just completed a study where we've retrospectively gone back and so Dave Thomas at the colony has been taking plasma and fecal samples of his cats for the last 10, 11 years, and now we're retrospectively going through and assessing changes in the microbiome composition, changes in its function using shotgun sequencing, and then overlaying that with changes in the lipidome, which is the lipid metabolites in plasma and lipid metabolites in feces, to try and understand if we can see shifts with age, and then understand what those shifts might mean. Ultimately, it would be great if we saw shifts that could be improved by diet, and therefore we improve the health of the aging cat. So that's a piece of research that we're sort of ticking away with now. In terms of the results, we see shifts in the microbiome, and we see shifts in the associated with age, and we see shifts in the function of the microbiome associated with age. But what's really interesting is that those shifts are greater in different diets. So the cats fed the retorted diet are you do see an age effect, but it's quite small, whereas cats fed a kibble diet, there's quite a large variation in those age effects. It's a little bit hard to explain without a PowerPoint. Could be their commercial diets, and there are macronutrient composition is relatively consistent across the lifespan, but there will be differences in specific ingredients and potentially associated with that differences in some of the more what's the word? Micronutrients.
Nutrients? Yep.
So there might be changes in the micronutrient status, of course. But it's certainly interesting. And one of the challenges we have when we're working in a research context, you have colony animals, limited number of animals. But there was a nice paper put out by Karina Salt and her colleagues that accessed or utilised data from one of the Banfield Veterinary Hospital, I believe, where she's gone through and she's used big data tools, so really good bioinformatics and things like that to understand those shifts in ages, that shift in health as the animal ages. So I think going forward, we've got this ability to look at big data and what those trends are saying, and then answer some of those questions using discrete research experiments. But certainly her data suggests that about 8 years of age, there's a shift in sort of health status in the cat, and our data shows that you see changes in macronutrient digestibility and changes in the microbiome, changes in the lipidome and the metabolome at about that same age. So there's something happening at around 8 years of age, which appears to be that switch from healthy adult to perhaps unhealthy elderly or more senior animal.
Right. So what we're really talking about is something related to the physiological aging process. And when that aging process starts to impact the animal's ability I'm just going to use process the diet as a kind of large catchphrase.
Yes.
One question that's fairly relevant to the industry is that you mentioned that there's a lot more diversity, there's a lot more change in the animals that are fed the extruded. And I was mainly wondering, because it's come up recently in a few other podcasts and in conversations, the fact that crude fiber, and as we move away from crude fiber being the reportable and towards TDF also includes resistant starch. Mm-hmm. And if companies aren't measuring starch cook as an example, are we potentially talking about how TDF has been likely has been swinging and far more variable in extruded diets? And I don't know enough about cooking, but is retorting a lot more consistent in its cooking process than extrusion would be, as an example? And I don't know that.
Yeah, so it's a good question. Certainly, consistency with how we measure things is always important when you're trying to understand long-term consequences of anything. With retorting, the big difference between retorting and extruding, and I don't come from a food production background, so it's quite high level here. The retorting process, you don't need carbohydrate, so you don't need that gellingness that happens. So canned diets, retorted diets, tend to have fairly minimal carbohydrate. What's in there tends to be sort of gels, etc., to kind of create the gravies. Whereas your extruded diets, you have upwards to almost 25, 50% in some cases, starch sources, because they need that They need that starch to physically extrude the kibble. So that's why it's we can't compare.
It's really tricky. There's far more carbohydrate in extruded. Yeah. Very good point.
Yeah.
So another question I have for you based on that comparison is Does water intake affect the microbiome?
I read that somewhere. You would assume yes. So certainly, if you're constipated, which is your bowels aren't moving, the stools are getting firmer and firmer, therefore drier. And there is certainly some shifts in the microbiome associated with that state. I would assume that there would be shifts. Haven't studied it directly, but I would assume that there would be some sort of shift there. Would it have a direct effect? I'm not sure. That's a good question, but certainly, if you've got if effectively you've got a firmer stool which is taking longer to pass versus a more softer stool. Oh, I love talking about poo. It's only 8:30 in the morning and I'm getting to do it. And there will be shifts there. So it's a good question. Not something I've seen any results on, but it would certainly be a good question from probably more from a constipation point of view than a nutritional point of view.
Yeah, I think we sometimes we don't focus enough on water either, despite the fact that if you have a deficiency in water, that'll kill you quick. Everything else is everything else is a lot longer to show up. So it's an interesting paradigm. Okay, so kind of coming back a little bit more, and the dog, the cat. So we've talked about life stage differences. Now, one thing that maybe pet food companies may or may not need to know, I would say they need to know almost everything that we talk about on this podcast. But when we think about shifts in microbiome, is it related? And so we're just going to talk about poo more here, Emma. Will there be a noticeable sign when you're picking up poo if you own a dog, or you're scooping out the litter box if you own cat? How far does that shift have to go before the owner notices?
So I don't think we know enough about the microbiome, personally. I think we, as scientists, for a variety of reasons, are very superficial in our interpretation of what's actually going on. So we tend to feed diets, and we see shifts, and we report on those shifts, but we don't actually know what they mean. We've really got no idea. So I'm going to, that to me is my frustration, because we're we're doing a lot of work, we're collecting a load of data, but we're not really interpreting it with any depth. And so the answer is I don't know, because we don't even know really what the microbiome of the cat and dog, what shifts are appropriate or what they mean for the health of the animal. If we look at sort of, I guess, a disease state, and the work that's being done out of Texas AMU is gold standard here. We know that dogs with and cats with IBS, irritable bowel or something like that, they have quite a different microbiome, and there appears to be some sort of dysbiosis where there's overrepresentation of specific groups. Those dogs and cats also have very runny poo, or potentially kind of there's a lot of variation. Sometimes it's okay and sometimes it's not. Certainly the microbiome has the ability to impact the smell of the feces, and that's through the breakdown of proteins, usually that are associated with those sort of more odorous compounds. Again, when we look at changes of diets, and we assess, spermidines and indoles and things like that in the feces, we report a statistical change, but we don't actually know if there's a biological significance to that. So we don't do sniff tests. So I did actually approach postulate this to the postulate an approach to the metabolomics lab. We had an ag research about how could we assess smell, and he said, the scientist there was, you can do it because, it's been done for things like wine and cheese and butter. But if you want to understand numerical shifts and how that affects, basically someone's going to have to sit there with a canister of poo and smell it and tell you. And it was like, I don't know if I would even do that to a PhD student. The short answer is that I don't think we know enough, but certainly there's a load of anecdotal evidence that suggests the less processed your diet, the firmer it is, the easier it is to pick up, and the less smell it has. That's largely anecdotal, and we don't know if that's due to the microbiome itself or the fact that these minimally processed diets tend to be more highly digestible, and so there's actually less fecal bulk there. So it's again a question where I think we just don't have enough information. And given there's a lot of marketing around my diets, do this, my diets do that, because we've seen shifts in microbiome, I think we're doing ourselves a bit of a disservice. We need to really be understanding what those shifts mean. So, yeah.
Yeah, no, great guidance and you're right. A lot of the time we run with something new in science, and part of that's because you start using new sexy words in your grants, and you're pursuing certain amounts of certain kinds of funding, or it becomes the hot topic in the industry, and everybody is pursuing gut health right now.
Exactly.
So it's part of that. So let's talk a little bit about, because you've alluded to this, and I thought maybe you could describe it a little bit better, how the digestibility of both the available or enzymatically digestible carbohydrate fat fraction and the protein fraction might impact what is flowing into the colon of both the dog and the cat. And if you could maybe for everyone online to describe when a total tract digestibility makes sense and when it doesn't with regard to these two.
Okay, you're really diving into my undergrad knowledge here. So in reality, total tract digestibility is basically looking at what you feed to the animal and what you collect in the poo. And that's our reality. It's very it's low invasive and easy to collect. When you look at total tract digestibility, however, what you're not considering is the protein contribution from the microbiome and from the cell, the intestinal cells, which slough into the total tract, into the digestive tract during the process of digestion. So there's, I guess, a methodological limitation, but it's our reality, because the alternative is you need to somehow obtain samples from the ileum and other parts of the digestive tract, which are highly invasive and things we don't actually want to be doing. In terms of looking at high carbohydrate or high protein, obviously if you've got lots of carbohydrate going into your diet, when that gets digested through the stomach and reaches the small and then ultimately the large intestine where it's fermented by the microbiome, you're going to have a lot more carbohydrate or a lot more kind of carbohydrate type molecules available to the microbiome there. From the point of view of, I guess, more omnivorous animals or herbivorous animals like so humans and cows and those sorts of things, those carbohydrates are fermented into short-chain fatty acids. There's a bit of a process going on, which are then absorbed by the host. In terms of high-protein diets, you get, obviously the same thing sort of happens, and you get an accumulation of, I guess, more proteinaceous substrates available to the microbiome to ferment. So when you look at the relative concentrations of short chain fatty acids in a carnivore versus a human, there's a lot less butyrate. However, and so the assumption, or has been for a long time, is that high protein diets decrease butyrate because and therefore are bad to are not good, not necessarily bad, but not good. But certainly with the work we can now do in the microbiome, we're understanding that actually there are bugs that can utilize the protein to make butyrate. And so, so it's not as simple as you need carbohydrate because you need the butyrate. Actually, the microbiome of the carnivore can actually utilize protein to make butyrate. And so we're seeing that there's a little bit of a shift from traditional butyrate fermentation pathways from carbohydrate to butyrate fermentation pathways from amino acids. So that's quite an interesting finding that we're sort of trying to tease out. And again, slight differences between cats and dogs. Definitely a diet effect and potentially a species effect there too. So, and then the other thing that obviously fermentation of proteins or amino acids or peptides are associated with your odorous compounds, as I mentioned earlier. Interestingly, we tend to think of, we've tended to cluster indole kind of quite, as a bad molecule, but there is research that suggests that indole, and not necessarily the indole derivatives, can actually improve gut barrier function in vitro models. So it's potentially that indole isn't necessarily the bad molecule it's always assumed to be. And there could be some nuances there. So that's sort of something that I'd like to sort of try and tease out in the future if I get the opportunity. Yeah. So more higher carbohydrate diets tend to have higher carbohydrate substrates, and you get that rise in short chain fatty acids, but actually might not be as dramatic, but you certainly see the ability of the microbiome to produce butyrate from protein ingredients too. So, yeah.
Which might be another step change that the cat uses to maintain its health and metabolism in essence. Okay, so we've talked about the microbiome. We've talked about life stage, and we've talked a little bit about the substrates that the microbiome is using. But let's now ladder to other parts of gut health, because I think a lot of the pet food industry, and like you said, we're making these broad sweeping conclusions based on microbiome shifts. But there's other metrics of gut health that are quite important to consider. And I know that you and Dave Thomas are looking at some of these. So do you want to describe some other gut health metrics that people may want to look at when they're looking at microbiome shifts and how they affect those other outcomes of gut health?
Yeah, sure. So if we take a step back into why we care about the microbiome in the first place, and at the start of this podcast, we talked about its role in the immune system and as part of a normal, healthy gut function. So certainly, the microbiome or the gut. It has its microbiome, it has its immune system, but it's also a physical barrier between the inside of our bodies and the outside world. And kind of bringing in this idea around at a certain age point, the cat loses its ability to deal with the excess nutrients, starts to put on weight, which is kind of what we've learned through this sort of aging study. We've sort of started thinking about why that might happen and why do we see these shifts in macronutrient digestibility associated with age. And so we've started to look at intestinal permeability as a marker of gut health and gut function. It's pretty, I won't say it's pretty easy. I don't think our PhD student would agree, but it's a relatively low invasive procedure. It's sugar probes. You dose the animal with those, and then you can measure them in urine or plasma, depending on what's easier to collect. And from those you can understand whether you've got a leaky gut, so whether your tight junctions are like this and the bad stuff can go through, or whether you've got a really nice solid intestinal wall. So that's one aspect we're taking. The other aspect which we kind of we think about, but if you're happy with your dog's or cat's poo, and you're not, it's good consistency, it's good, relatively good odor, it's always going to smell. Those sorts of things, you can pick it up easily. That's probably the best marker of, your dog or cat's gut health, right? If things are going in and they're coming out looking good, and your dog's got it's got a nice coat on it, same with your cat, you can be reasonably assured that the diet that you're feeding is actually working well for your animal. So yeah, never underestimate the value of nice feces.
Yeah. So on that, I couldn't agree more. There's so many places and jokes that you can go on this, and I certainly try to lay them down on undergrads, and they don't laugh that much when I talk about poo. But I know it's so disappointing. I do remind them that you cannot be a nutritionist unless you're cool with poo. And I'll make one caveat. I'm not okay with human poo.
Yes.
There's no way that I'm collecting human poo, but I don't have a problem with collecting animal poo. Yeah. And I just wanted to point out that I think that horse poo actually smells quite amazing.
I'm a fan of horse poo. Yep.
Yeah. Great horse poo. But jokes aside, to your point about consistency, is there a breadth though of healthy consistency? So, if a animal has pretty consistent but soft poos, is that healthy or not healthy, in contrast to the dog beside them or the cat beside them who has slightly firmer poos that are easy to pick up? So where on the spectrum of poo consistency is healthy? Is there a range, or is it really targeted?
So there is a scale that we use to assess fecal score or fecal health score. I think it's the Waltham poo scale that we've used, and they have a range. They range from 1 to 5, where 1 is very liquid and 5 is very, very hard. So the healthy range in that scale is somewhere from like 2.5 to kind of 3.5. So there is a scale, and within the scale there's a scale. So we know that there is quite a lot of variation in how individual dogs and cats deal with their diet. You would have over the years, and myself and pretty much anybody in pet nutrition would have dealt with my cat runs on this diet, struggled for years to try and help there, and obviously that's where the work of Texas AMU is really important. They're kind of understanding that kind of dysbiosis and irritable bowel kind of thing is. But I would say if your dog is soft, but consistently soft, and you're happy with him, he's got the right amount of energy, his coat is nice, he seems happy, then it's perfectly fine. If your dog or cat is consistently at the firmer end of the scale, but you're happy with that, and there's no issues with straining or constipation, those sorts of things, then that's perfectly fine as well. I think it's, we do, there is a lot of individual variation between, cats and dogs in terms of how they react or respond to different diets. So, I think there is,
Yeah, and you mentioned you mentioned as an example gut permeability as a measure. Can you outwardly have a dog or cat that is asymptomatic if you used fecal score, but actually has increased gut permeability?
Can you ask us that in 6 months' time?
I will. I guess it's interesting because one might precede the other one. And I think to your point about how little we know, when you're trying to piece these things together, you need that timeline to understand whether the chicken or the egg came first. Do you start to get microbiome shifts before you get gut perm shifts, or do you get gut perm shifts that then underpin microbiome shifts? And then where's metabolism affected along this cascade? So we don't have those answers, but you and Dave are working hard to provide those to the industry.
Yes, we are. Yes, yep. So I think any fundamental knowledge that we can get around the aging process in terms of gut health and permeability, the microbiome, will basically it's part of the puzzle, right? And certainly, when we've got cats living upwards of 16, 22, I think is some of the older ones I've heard of, years of age. We want our cats to be healthy when they're that old, not kind of struggling, not scraggly. We want them to be enjoying their life, and so the more that we can understand around what's actually happening in that time frame, and can we improve it with dietary nutrients, then that's the long-term goal of this research. Of course, you don't have all the answers straight away, but that's the goal.
Yeah. We want to increase health span, not
health span. Yeah, exactly. That's it perfectly. Yeah. There is a focus on lifespan, and but actually, it's the health that is associated with that that's really important. And, in our older animals, the older you get, the older they get, the more they experience disease. So things like chronic kidney disease in cats, osteoarthritis, those sorts of things. There are gut links, microbiome links to those sorts of diseases too. So I suspect the more we understand what's happening in a healthy aging, an otherwise healthy aging animal, the more we'll be able to utilize that information to try and help with those real specific disease states that we see in our aging animals.
Yeah, I think a lot of definitely our pet owners and don't understand it as well. But I think most people don't understand that every mammal on the planet actually contains more other organisms in the form of bacteria than the actual cells that our own tissues possess and accumulate. So we have this crazy symbiotic relationship. So the question is, when does that start to go to crap? And lead to deterioration and aging. So I think everybody's going to be really looking forward to that work. And not only will it impact pet nutrition, but it's going to drive curiosity in other animals, including humans who we age with.
Yes, exactly.
So maybe to bring it together on this, when we think about maintaining that healthy microbiome, and we know that we still have a lot more to learn, if you base it on what you know today. How would you advise companies to formulate the difference between adult maintenance formulations and let's juxtaposition it pretty far, go with a super senior formula for both dogs and cats? How might those look a little bit different? And maybe it's not the composition, maybe it's thinking about other things like probiotics and prebiotics and postbiotics and a combination of all three.
Yeah. So interestingly, when you look at things like AFCO and FEDF, which is the European nutritional regulation, there isn't any differentiation beyond pregnancy and kitten or puppy and sort of adult. And adult is basically typically anything from 1 year of age, both cats and dogs. So I think the first thing we need to do is really understand what our senior cats and dogs need in terms of specific nutrients. We know that their needs are different. There is a lot of work, especially I think in the more veterinary side of things rather than, say, animal science around older dogs needing more protein. But that's not reflected in our regulatory guidelines, right? We know that if you supplement old cats with various probiotics and prebiotics and long chain omega-3 fatty acids, for example, they are healthier or they live a little bit longer. So we know that if those nutrients have the ability to improve health span, then it suggests that there's actually a greater need for those nutrients, right? So I think the first thing we need to do is really understand. So I think we can, with relative certainty, say it's for the cat at around 8 years of age, there's a shift that happens. So what are the nutrient needs post that? And then I would like to see the likes of our regulatory bodies go, we have a growing pregnancy lactation, we have a young adult, so 1 to 7, and then we have a senior recommendation. And then given we have cats living anywhere from 12 to 22 years of age, we probably need some work in that super senior space as well. So in terms of formulating formulating diets, at the moment we're limited because our AFCO regulation is very broad, and I don't think we understand the nutrient requirements of healthy adult cats as well as we should, let alone what our more vulnerable elderly populations of pets will need as well. So given that's our limitation, I think, from a formulation point of view, I think pet food companies should be looking at what work has been done in supplementation of older animals, what sort of benefits they see, the use of prebiotics. I'm a fan of prebiotics rather than probiotics at this stage. That's another discussion. Building up the good bugs that are naturally present, naturally and I think naturally is the right word. Present in the carnivore gut. To really understand what we should be putting in our diets for our senior animals. So I think, I guess in summary, there's a lot of work that needs to be done given our pets are living such long lives with us now, and I think we have an obligation to do them better and get better data and get that data regulated, or this is what we must be doing now.
Yeah, I couldn't, and definitely I'm sure all of our pet food industry listeners have heard it from me time and time again. This really speaks to the industry coming together to support basic research that would not benefit any single company, but benefit all pets.
That's right, yeah.
And this is important because there is not in a lot of, well, in most countries, there isn't remotely as much funding for pet, if there is any at all.
That's right.
In contrast to humans and agricultural animals. So just a plug for that.
Yeah.
So
I think the contra to that is also true, and I've sort of alluded to this in the past, where the pet food industry used to lag behind the human industry. It was 5 years, or it was 10 years, and it was 5 years, in terms of health nutrient trends or ingredient trends. And then it was sort of 18 months, and now it's almost instantaneous. If a human is eating brown eggs, it wants their cat to be eating brown eggs too. And the industry shifts to those demands without actually thinking, is this the right thing for the animal? And I think, pardon?
I couldn't agree more.
So I think that as a science community and as a pet food industry, we should be saying, hey, let's look at this better before we implement it. And the DCM debacle is case in point. We shifted, and there was a whole mess, which is sort of we're crawling out of the weeds now. But yeah, I think in terms of humans imposing their desires on pet food, we should be saying, actually, your dog isn't designed to eat, I'll say blueberries because that's my favourite one to talk about. They might actually be healthy, but that's not the point. It's not designed to do that. So let's just look at some research first. Let's understand the consequences of feeding that rather than just including that because it's a trend. So I think that's probably the challenge for us all going forward, is actually kind of asking pet owners just to let's get some research before we do this. Yeah.
And then reminding the pet food industry too that if they're going to do a step change, they need to put the research behind it before they launch that, because
that's
they're
right.
Afterwards.
That's right. Yes, totally agree. Yeah.
Couldn't agree more. Well, thank you for the science discussion today. But to end off, because I personally find you quite a formidable female leader in the pet food industry.
Oh, wow.
Yeah, you're pretty cool. I was wondering if you wouldn't mind departing some advice on especially young women in the pet food industry, and maybe one thing that you would highly advise that they consider and one thing that you highly advise that they avoid.
Okay, that's thank you for your glowing reference there. So I think the thing I would advise is to take every opportunity. Don't think you can't do it because you haven't done it or because you've only done maybe a little bit. Back yourself and take the random opportunity. Have the conversation, maybe with a startup who is thinking about things in a different way. Never be afraid to say you don't know. I think there's a lot of, we try and bluff our way through things, but I think there's a lot of power in being honest and our brains all work differently. If you need more time to reflect on something, then say that, right? Don't be afraid not to speak up because you don't have the answer. In terms of, those would probably be the same things. Take the opportunities, especially the random ones. I think, given now the importance of things like communication in every industry, the ability to speak, upwards to management or boards, downwards to maybe people in your organisation, sideways. Communication papers or marketing papers. That's my biggest regret, is I didn't sort of delve into that more. to understand, what they're trying to achieve from their perspective. So I think that's probably what I would, and what I do suggest to PhD students that I mentor is, the more perspectives you can get on anything, the better the question you can ask, and then asking questions is what's going to solve the problem. So that's what I would suggest, recommend. Yeah.
Okay. Do you want to say one thing to avoid, or do you want to avoid that question?
I'm going to say avoid putting yourself in a box. So as scientists, we tend to think of ourselves as scientists. So I'm a scientist, and that's what I am. But actually what you are is a problem solver. You're a communicator. Sometimes you're a contract writer, business development manager. You're a whole heap of things being a scientist in the reality of the scientific world today. You're not just at the lab bench pipetting, you're problem solving, you're adapting, you're innovating. So mine would be avoiding putting yourself in a box and sort of thinking about the skills you have in that wider context, and that can be uncomfortable. And so I would suggest to talk to somebody you respect, whose opinion you respect, and sort of if you're struggling to figure out what it is you can bring to the table, talking to them, and thinking about you're a time manager, you're a project manager, you're all these things. You're not just a scientist. So that's what I'd say.
Of skills. Yeah. And it's lifelong development, right? So they have to think.
That's correct. Yeah. Yeah. We're always learning. Yeah.
Always learning. Absolutely. Well, with that, Emma, thank you so much for joining me today. And I look forward to talking to you again. There's other topics that I have slated for as well. Energy being one of, we talk about that one forever. So stay tuned for that if you're a listener. And thank you, Emma, for joining us today.
Thank you so much for having me. And thanks everybody for listening.